we set up a platform for the non-invasive identification of developmentally competent or incompetent mouse oocytes during their maturation from GV to metaphase 2. This platform is based on three main ingredients, or how they call them, algorithms. A first algorithm extracts the most salient information from an image, and this is called the saliency map algorithm. The most salient information of an image means that the image is made lighter by removing all known essential information to be able to identify that object. When I was younger, I used to be a bird watcher, and I do remember that I could identify birds just by looking at their essential characteristics, features like the movement of the wings, or the way they were singing, for example. So the saliency map algorithm extracts the most salient information. But once this information has been extracted, we need to maintain it, to follow it, even during movement. And to do this, we use the binary robust invariant scalable key points algorithm, or BRISC. And this keeps track of the information while this is moving from one frame to the other. A third set of algorithms direction of our information. So pulling together all the information from these three algorithms, we set up a platform which begins with the acquisition of time-lapse images during the 15 hours of GV to M2 transition. And frames were taken every 8 minutes for a total 112 frames. In the next slide, we can observe a time-lapse movie of an SN oocyte during the GV to M2 transition. And please notice the timing of polar body 1 extrusion. And PIV records movement by comparing the speed at a time point with that of the previous one. And our next slide shows an SN oocyte during the GV to M2 transition. And notice how this type of oocyte extrudes the polar body later than the SN oocyte. We analyzed a total of 127 SN and 109 NSN oocytes, and found that they are significantly different at 4 time intervals. Importantly, whenever we restricted the analysis to only one of these time intervals, or to a combination of them, the differences between the two profiles at this time point disappeared. Also, when we analyze the profile of single oocytes, which is what we need to make in the clinical a prediction, we could not tell which was which. This is why we implemented our analysis with a feed-forward artificial neural network, which was trained first with known samples. So we told the system that the frames it was looking at were belonging to an SN oocyte or to an SN oocyte. Then the artificial neural network, which is named among the many that are present in the artificial intelligence set of tools. This is what's called feed-forward artificial neural network, and it was tested blinded. So the results showed that our platform could detect the developmental competence of an oocyte with a 91% accuracy, which is comparable to that obtained with a well-trained operator. And actually, it's actually a bit slightly higher. So an operator usually has an accuracy of about 86%. With this in mind, we tried our platform on human oocytes and on human preimplantation embryos. And these time-lapse movies show you the analysis of two groups of preimplantation embryos. One group of embryos that reach the blastocyst stage, like this one. And please notice here the spikes of the first, second, third, and fourth cell divisions. And this one is an example of a group of embryos that arrested development sometime before blastocyst. We analyzed in the next slide about 150 embryos throughout the whole preimplantation development, 150 embryos that reached the blastocyst stage and 150 that did not. And at first we decided to concentrate only on the first two cell division time intervals. And you can see here the two profiles, the blue one of the embryos that reached the blastocyst stage, and in red, the embryos that did not reach the blastocyst stage. Now, the two profiles are very similar, although at these time intervals, so before the first cell division and at the second cell division, they are differentially, they show a difference that is significant. Of course, this is when, again, as we did with the mouse, this is when we group the two types of oocytes in one single comparative result. But we could not detect the difference between each single embryo. For this reason, we implemented these analysis with an artificial neural network. This is another type. If you remember, before we used the feed-forward artificial neural network, this is called a long short-term memory neural network, which was capable of, and you can see here in the next slide, of highlighting a difference between the two types of embryos, the developmentally competent and the incompetent. Differently from the mouse, The human analysis with this artificial neural network reached the accuracy that we obtained with a trained embryologist. But when the analysis with the artificial neural network was combined in one single analysis with that obtained from the experienced embryologist, our capacity and accuracy to identify during the first 2 cell cycles of the pre-implantation development, the developmental competence of that embryo was much higher and significantly higher than that of the operator alone. So what we are, what our work underlines, although this is still a proof of concept, we show that combining artificial neural network analysis with that of the operator, which still remains important, we can obtain an important good advice on how to classify as developmentally competent or incompetent our very early embryos during their pre-implantation development. Of course, this is a proof of concept study. We need to reinforce our data, increasing our sample of embryos. And of course, we need also to compare the outcome between different instruments that are used in the clinics. We used the biostation for mouse studies and the embryoscope for human study. And of course, we will need to compare our results with those that could be obtained with our setup in other laboratories. And I thank you for your attention. And before I end, I would like to thank our collaborators from the 9.baby clinic in Bologna, Giovanni Coticchio, Raffaella Sciaino, and Andrea Borini, the Department of Engineer and Bioinformatics of the University of Pavia, Professor Bellazzi and Dr. Nicora, and my laboratory of developmental biology at the University of Pavia. Thanks again for your attention.
Okay, I would like to thank both speakers for their really focus in the full of information talks. And I can see there's a lot of questions, and the live competition is being led at this moment by a question by Thomas Eberton. So we're going to start answering the question relevant to the first talk, and then we will move on to the second. In doing that, we have at least 15 minutes, so plenty of time to do that. So the first question from Dr. Thomas Ebner, Austria. So his question is, well, after thanking the speaker, he would like to know how would the speaker explain the fact that cytoplasmic waves originate from the male pronucleus, perhaps outgrowth of microtubules. Yanis?
Yes, thank you, Thomas, for your question. This is a very good question. It's actually not easy to answer. Certainly, the time-lapse study by Giovanni Coticchio showed that the male PN has a central role in determining fertility events during fertilization. So it's not just the direction of the waves, it's also the timings of the position of PN juxtaposition and timing of disappearance and all that. So I think that we can postulate that the male pronucleus so the sperm, okay, the position of the sperm has a role in regulating the early cellular events. We have to remember that the sperm is the carrier and the carrier of the first microtubule and the form is responsible for the sperm aster. So the sperm aster creates the first network of microtubules within the fertilized oocytes, which brings the male and the female pronuclei together. So certainly cytoskeleton has a very important role in these early events, and the rearrangement of the cytoskeleton can possibly be regulated by the sperm and the position of the sperm within the oocytes. I'm not aware of any more specific data. There might be evidence out there, but it seems that the male pronucleus plays a role.
Okay, seems to be a very exhaustive answer. Thank you very much, Yanis. We can move on to the second in list in the league table as a question panel point, which is made by our chair, extra chair, Cristina Magni. So again, the next question to Yanis. Quote, do 3PN embryos have different time-lapse patterns compared to 2PN ones? And you also mentioned that 1/3 of them are euploid. Any evidence that they are not parthenoids? Yanis?
Yes, thank you, Christina, for your question. I'm very happy to answer it. It's very possible that the diploid 3PNs could be parthenoids. So this study that I showed in my presentation was actually based on a very small number of oocytes. So it very well may be that parthenogenesis is involved. Now, regarding the morphokinetic characteristics, there are studies out there showing that 3PN oocytes have different morphokinetic characteristics compared to 2PN oocytes. The most important characteristic with 3PN oocytes is that they have a larger proportion of zygotes goes on to cleave directly to 3 cells. And that is the case both for 3PNs that were fertilized by conventional IVF and also fertilized by ICSI. So yes, it has been shown that 3PN zygotes can cleave, they can develop. They have a lower blastocyst formation rate. They have various different morphokinetic timings, but most of all, they have a higher incidence of direct cleavage.
Okay, thank you very much. We have time for at least another question relevant to the first talk. And the third best voted question is from Maha. And the question in particular is that is relevant to the possibility that denudation, in other words, mechanical manipulation, can affect somehow distribution of pronuclei, or in general, I would imagine, the entire choreography of fertilization. Yanis?
That's a good question. It's the denudation, and also the actual procedure of injecting a sperm inside an egg is totally different compared to normal fertilization by the sperm. So we have to remember that ICSI circumvents all the barriers that the sperm has to cross in order to get inside the egg. So one very important event that happens during fertilization, and also during conventional IVF, is the fusion of the membranes of the sperm and the egg. So there's a lot of evidence out there that this fusion is the combination of very important cell surface proteins, okay, which triggers a whole sequence of events within the egg, including the cortical reaction, including the block to polyspermy. So it has been shown that ICSI can lead to a slightly modified sequence in the cellular machinery. So it could be possible that it might affect the pronuclei as well. However, we have to bear in mind that the majority of time-lapse studies studying early events involve fertilization with ICSI. So using ICSI is the only way that we can actually study the very early fertilization events that would otherwise be lost if we had fertilized the eggs using conventional IVF. So I think this is a factor we have to recognize. Okay, let's all studies use ICSI.
Okay, thank you very much, Yanis. That was fantastic, because also unwillingly, perhaps also because you have a crystal ball, you were able to ask another question which was relevant to the possible impact of ICSI. So yeah, you got one pigeon with one stone, two pigeons with one stone. Excellent. So I would like to then ask my colleague and co-chairperson, Maria Louise Grondal, to move on with the question for the second talk. Thank you very much, Maria Louise.
Yeah, and there is a question to Mauricio from Sangi Gonzalez, and it is: has your team correlated AI features to components of the ooplasm, and can the effect of sperm be quantified in the models based on an image of an oocyte?
Our system picks up all the changes in movement that occur within the maturation or developmental drop, where you culture, you mature your oocyte in the case of the mouse, or you grow your embryo with the experiments that we do with human. So the system picks up all the movements that occur within that drop. And of course, most of the movements are due to the presence of the oocyte, which is maturing, or of the embryo that is developing. And most of the movement are cytoplasmic movement. So anything that can be seen by the algorithm is picked up. Whether it's gonna be interesting or a key feature for a classification, that is just a matter of trying and see what happens. Of course, during maturation, you have the passage from GV to metaphase 2. So you have big changes that occur, and as well as when you watch what happens during embryo development, you have the first, second, third, in our case, fourth cell division. So you have big changes that occur within the ooplasm, and those big changes are picked up. Although, as I said with the mouse, those big changes alone are not enough to make a classification. When we concentrated on what happens at the time of those big changes, if you remember my slide where I showed the four main maturation windows that were that picked up a significant difference between the two types of oocytes. Those windows alone were not enough. So something else is picked up in between. And this is probably what occurs with the human embryo as well. Whether we can take advantage, we can include the information that we might be able to record after fertilization or ICSI. That's a matter of trying. We haven't done it yet, but this is something we would like to do. Yeah.
Okay. Thank you. And question from Ursula Ritter. First, she wants to congratulate you on an excellent presentation. And then she asked, would it be possible to employ the AI also in the case of IVF and not only ICSI? And we have touched upon it before.
Yes, it would be possible in the case of IVF as well. What we did was to record the changes after fertilization. So if Ursula is thinking of analyzing what happens after in vitro fertilization, I think that's possible. Yes, but our goal is also to include the recording of what happens at the time of fertilization, whether this occurs with IVF or
and then we have a question, I guess it's for both speakers. What is your recommendation for laboratories without time-lapse technology in terms of fertilization evaluation and embryo monitoring during culture? Can we learn something from your observations?
Could you repeat the question?
Yeah.
I didn't get it.
I can try to answer, although it's not easy to answer right now. As I said, with our experiments, so far we analyzed only a small group of embryos. What we are trying to do is to set up a sort of guidelines on how to record the videos. Of course, not all the videos that we first analyzed were good for our further analysis, because maybe they haven't been done correctly, or so we are trying to set up some sort of guidelines in order to provide us with many more embryos movies, and to be able to...
Sorry, my connection is quite bad here.
Sorry? Sorry, Marie-Louise?
Oh, I'm sorry, I didn't hear all of your answer. My connection is not that good.
Oh, right, sorry.
Should I take the next question?
Should I answer again?
Yeah, no, it's, I guess we go to the next question then. It's from Anne-Maike Melker. It's regarding the timing of division. Is it dependent of culture medium? Have you had to change to look into the effect of culture medium for your...
I can answer that, yeah. Sorry. There have been a few studies performed looking at there have been various studies in the literature looking at morphokinetic timings in different culture media, and mainly looking at single versus sequential media. So it looks like although the evidence is rather inconclusive, I think the answer is that there do not seem to be big differences in morphokinetic timings depending on the culture medium. There might be some studies identifying, for example, a shorter T5 in a single step medium compared to sequential, but overall some studies find some minor differences, other studies find no differences whatsoever. So still inconclusive, but it doesn't look like the medium has such a big effect, according to current evidence.
Marie-Louise, we have 1 minute if we manage to get another quick question and answer.
Yeah, I have a question here with kind of also some of the questions from the audience. It's regarding the correlation between the early observations and then euploidy, and not blastocyst development. Did you have a chance to look into that, Mauricio?
No, not yet. Sorry.
Okay.
But it's a very interesting point, of course. Yeah, but we haven't done anything yet so far. Yeah.
I guess this is. Are we out of time now, Giovanni? So then I will thank both speakers for the excellent presentations, and I will thank the audience for the good questions, and then I will close the session.
Thank you very much.
Thank you very much, you all.